which the following are the more important. 1. A striatum of white fibers covering the superficial aspect of the molecular layer, plexus of Exner. 2. The band of Bechtereau in the outer part of the layer of small pyramidal cells. 3. The band of Jenneri, or external band of Ballinger, running through the latter of large pyramidal cells. 4. The interior band of Ballinger, between the layers of the large pyramidal cells and the polymorphous layer. 5. The deep tangential fibers in the lower part of the polymorphous layer. The tangential fibers consist of A. The collateral of the pyramidal and polymorphous cells and the cells of Martinati. B. The branching axons of Golgi's cells. C. The collaterals and terminal arborizations of the projection, commissural, and association fibers. The radial fibers. Some of these, that is, the axons of the pyramidal and polymorphous cells, descend into the central white matter, while others, the terminations of the projection, commissural, or association fibers, ascend to end in the cortex. The axons of the cells of Martinati are also ascending fibers. Special types of cerebral cortex. It has already been pointed out that the minute structure of the cortex differs in different regions of the hemisphere, and A.W. Campbell has endeavored to prove, as the result of an exhaustive examination of a series of humans and anthropoid brains, quote, that there exists a direct correlation between the physiologic function and histologic structure, end quote. The principal regions where the typical structures departed from will now be referred to. 1. In the calcarine fissure and the gyri bounding it, the internal line of Ballinger is absent, while the band of Jenneri is of considerable thickness and forms a characteristic feature of this region of the cortex. If a section be examined microscopically, an additional layer of cells is seen to be interpolated between the molecular layer and the layer of small pyramidal cells. This extra layer consists of 2 or 3 strata of fusiform cells, the long axis of which are at right angles to the surface. Each cell gives off 2 dendrites, external and internal, from the latter of which the axon arises and passes into the white central substance. In the layer of small pyramidal cells, fusiform cells identical with the above are seen, as well as ovoid or star-like cells with ascending axons, cells of Martinati. This is the visual area of the cortex, and it has been shown by J.S. Bolton that in old-standing cases of optic atrophy, the thickness of Jenneri's band is reduced by nearly 50%. A.W. Campbell says, quote, histologically, 2 distinct types of cortex can be made out in the occipital lobe. The first of these coats the walls and bounding convolutions of the calcarine fissure and is distinguished by the well-known line of Jenneri or Vic d'Azur. The second area forms an investing zone a centimeter or more broad around the first and is characterized by a remarkable wealth of fibers, as well as by a curious piriform cells of large size richly stocked with chromophobe elements, cells which seem to have escaped the observation of Ramani Cajal, Bolton, and others who have worked at this region. As to the functions of these 2 regions, there is abundant evidence, anatomical, embryological, and pathological, to show that the first, or calcarine area, is that to which visual sensations primarily pass, and we are gradually obtaining proof to the effect that the second investing area is constituted for the interpretation and further elaboration of these sensations. These areas therefore deserve the name visio-sensory and visio-psychic, end quote. 2. The anterior central gyrus is characterized by the presence of the giant cells of Betz and by, quote, a wealth of nerve fibers immeasurably superior to that of any other part, end quote, Campbell, and in these respects differs from the posterior central gyrus. These 2 gyri, together with the paracentral lobule, were long regarded as constituting the, quote, motor areas, end quote, of the hemispheres. But Sherrington and Grunbaum have shown that in the chimpanzee the motor area never extends onto the free face of the posterior central gyrus, but occupies the entire length of the anterior central gyrus, and in most cases the greater part or whole of its width. It extends into the depth of the central sulcus, occupying the anterior wall, and in some places the floor, and in some extending even into the deeper part of the posterior wall of the sulcus. 3. In the hippocampus, the molecular layer is very thick and contains a large number of Golgi cells. It has been divided into 3 strata. A. S convolution or S granulosum, containing many tangential fibers. B. S. lacunosum, presenting numerous vascular spaces. C. S. radiatum, exhibiting a rich plexus of fibrils. The two layers of pyramidal cells are condensed into one, and the cells are mostly of large size. The axons of the cells of the polymorphous layer may run in an ascending, a descending, or a horizontal direction. Between the polymorphous layer and the ventricular ependema is the white substance of the alveus. 4. In the fascia dentata hippocampi, or dentate gyrus, the molecular layer contains some pyramidal cells, while the layer of pyramidal cells is almost entirely represented by small ovoid cells. 5. The olfactory bulb. In many of the lower animals, this contains a cavity which communicates through the olfactory tract with the lateral ventricle. In man, the original cavity is filled up by neuroglia, and its wall becomes thickened, but more and more so on the ventral than on the dorsal aspect. Its dorsal part contains a small amount of gray and white substance, but it is scanty and ill-defined. A section through the ventral part shows it to consist of the following layers from without inward. 1. A layer of olfactory nerve fibers, which are the non-medullated axons prolonged from the olfactory cells of the nasal cavity, and reach the bulb by passing through the cribriform plate of the ethmoid bone. At first they cover the bulb, and then penetrate at the end by forming synapses with the dendrites of the mitral cells, presently to be described. 2. Glomerular layer. This layer contains numerous spheroidal reticulated enlargements termed glomeruli, produced by the branching and arborizations of the processes of the olfactory nerve fibers with the descending dendrites of the mitral cells. 3. Molecular layer. This is formed of a matrix of neuroglia embedded in which are the mitral cells. These cells are pyramidal in shape, and the basal part of each gives off a thick dendrite which descends into the glomerular layer where it arborizes as indicated above, and others which interlace with similar dendrites of neighboring mitral cells. The axons pass through the next layer into the white matter of the bulb, and after becoming bent on themselves at right angle, are continued into the olfactory tract. 4. Nerve fiber layer. This lies next to the central core of neuroglia, and its fibers consist of the axons or afferent processes of the mitral cells passing into the brain. Some efferent fibers are, however, also present and end in the molecular layer, but nothing is known as to their exact origin. Weight of the encephalon. The average weight of the brain in the adult male is about 1,380 grams, that of the female about 1,250 grams. In the male, the maximum weight out of 278 cases was 1,840 grams, and the minimum weight 964 grams. The maximum weight of the adult female brain out of 191 cases was 1,585 grams, and the minimum weight 879 grams. The brain increases rapidly during the first 4 years of life and reaches its maximum weight by about the 20th year. As age advances, the brain decreases slowly in weight. In old age, the decrease takes place more rapidly, to the extent of about 28 grams per year. The human brain is heavier than that of any of the lower animals except the elephant and whale. The brain of the former weighs from 3.5 to 5.4 kilograms, and that of the whale, in a specimen 19 meters long, weighed rather more than 6.7 kilograms. Cerebral localization. Physiological and pathological research have now gone far to prove that a considerable part of the surface of the brain may be mapped out into a series of more or less definite areas, each of which is intimately connected with some well-defined function. Motor areas. The motor area occupies the anterior central and frontal gyri and the paracentral lobule. The centers for the lower limb are located on the uppermost part of the anterior central gyrus and its continuation onto the paracentral lobule. Those for the trunk are on the upper portion, and those for the upper limb on the middle portion of the anterior central gyrus. The facial centers are situated on the lower part of the anterior central gyrus, those for the tongue, larynx, muscles of mastication, and pharynx on the frontal operculum, while those for the head and neck occupy the posterior end of the middle frontal gyrus. Sensory areas. Tactile and temperature senses are located on the posterior central gyrus, while the sense of form and solidity is on the superior parietal lobule and precuneus. With regard to the special senses, the area for the sense of taste is probably related to the uncus and hippocampal gyrus. The auditory area occupies the middle third of the superior temporal gyrus and adjacent gyri in the lateral fissure. The visual area, the calcarine fissure and cuneus. The olfactory area, the rhinencephalon. As special centers of much importance may be noted, the emissive center for speech on the left inferior